So good morning, everyone. Thank you for coming. Thank you also for those online. I would like to start by welcoming George. It's the first time that George Assis is in the museum. So let's welcome him for that. I'm going to have a few words of introduction of George. Some of you might know him, others not. He did his PhD in marine science in 2013. Right?
Yes.
He had a thesis on modeling marine forests. The title of the thesis was Marine Foundation Species, Shifting Ranges and Genetic Baselines. So it was crossing information on genetic patterns and species distributions, as I suppose he's going to do today as well. But he also had a BSc Licenciatura in 2005, also in marine biology and fisheries, where he had a thesis whose title was Rapid Assessment Protocol for the Implementation of Marine Protected Areas in Gran Canaria. So he has his experience in working in Spain.
Yeah.
He classifies himself as a marine macroecologist and data scientist. He also leads the Biodiversity Data Science Research Group at the Center for Marine Science, CCMAR, at University of Algarve. Also visiting professor at the Nord Universitet in Bodo,
Yes.
in Norway. And of course, an esteemed editor in ecography. Very important.
Trying to.
Yeah. It combines machine learning, AI, high-performance computing to investigate two intertwined questions. What mechanisms from genes to ecosystems govern the global distribution and abundance of marine life? And how past, present, and projected future climate change alters those patterns. So today is going to be talking about drivers and patterns of marine forest biodiversity in a changing climate, a macroecological perspective from genes to ecosystems. Thank you, George, for coming.
Thank you. Thank you so much, Miguel, for inviting me. Thank you for CSIC because, the infrastructure is amazing. Thank you for you guys for attending the presentation and those that are online. I hope so. Thank you so much. Well, this is an amazing introduction of myself. I think I'll keep this video and then forward for those that want to know me better. Thank you for searching my background. So as Miguel said, in our team, we are interested in exploring the drivers and the patterns of marine biodiversity. And today, using marine forest species, I'm going to showcase some of the studies that we do in our team. So this is just an overview of my presentation. I will start to briefly explain why we work mostly with marine forest species and what are they. Then I'll jump into the macroecological drivers that shape these hotspots with higher and endemic species richness at the global scale. Then I will go from species richness, meaning interspecific, to intraspecific, genetic diversity patterns at the global scale and their drivers. And then I'll say, or I'll show some studies on how range shifts of future climates might reshape these patterns of species richness and communities, and also the gene pools of these marine forests. And then I'll briefly make some notes on how this information can help on the prioritization of conservation, meaning the implementation of marine protected areas. So first, starting, why do in our team, or why do I have this background on marine forests? So for those that you don't know, these species, mostly composed, for instance, from mangroves and kelp forests, which are these amazing brown algae that can grow up to 60 meters of height, and seagrass meadows that populate the Mediterranean coastlines of Spain, for instance, and also the cold water corals that are distributed globally. They are these marine forests, and they are very important. So they structure these critical habitats that provide full food, shelter, and nurturing routes for an amazing amount of biodiversity that is associated. And many of these species that are associated are also of commercial interests, like the Atlantic cod. This group of species, these marine forests, they also provide key ecosystem services, like they protect our shorelines from erosion and storms, and they have a huge contribution on nutrient cycling, meaning that they absorb the excess of nutrients in the water, and they, through primary productivity, they produce organic matter. When this organic matter is trapped in the sediments or trapped in deeper waters, these marine forests have a huge role in carbon sequestration, thereby mitigating the effects of climate change. Okay. So another point that for us is critical on why do we work with these species is that they are sensitive to climate change. So they are shifting their distribution globally. So there are reports globally on collapses of abundance and even of their occurrence, inclusive in the Iberian Peninsula, around southern Portugal, where I live, or the Bay of Biscay, where many of these species have collapsed and they are no longer there. And most importantly, and for me as an ecologist, I like to work with them because of their sessile nature. So they are attached to the bottom, they are there, and they are sensitive to climate change. So it's a very good model to explore the relationship between the drivers and patterns of how we make this link between climate change and the changes in the abundance or the occurrence of species, because changes in the environment will directly affect what we perceive in the field. So it's very useful as a model. So the first part of my presentation is based on many studies that we did on addressing the global patterns of species richness of this marine forest at a global scale. So global biodiversity patterns, like these hotspots of species richness or hotspots of endemicity, or even the way the communities are structured in space, this is basically underpinned by long-term geological events and also climate fluctuations of the past. But estimating these global patterns is very challenging. I mean, mapping these hotspots of species richness and endemicity is very challenging. However, it is very important, not only for us ecologists, but also for conservation. And current knowledge is mainly towards terrestrial environment. And in the marine environment, what we have nowadays as estimates of these patterns is mostly towards fish and mammals. And there is no, or there wasn't, no overall view of the way these marine forests were distributed in space. So our team explored the drivers and the patterns across these groups, and mostly for these kelp forests that are the large brown algae, the seagrasses, which are these meadows that grow, for instance, in the Mediterranean, and also the cold water corals. So how have we made this? So we have used species distribution models. I think that most of you are aware, but for those that don't, basically what they do, they create this model relationship between the occurrence of presence records of our species of interest, and we relate these occurrences with the environment that they are exposed to, for instance, temperature and nutrient conditions, or even ice conditions in higher latitudes, and then the models are allowed to predict the distribution of species. Basically, the models capture the effect of the different environmental variables, for instance, the effect of minimum temperature, meaning that if we increase minimum temperature, maybe a given species will augment its probability of occurring in a given site, or if we increase maximum temperature, is detrimental physiologically, so maybe we have a decrease on the probability of occurrence. So the model learns these functions, and then we can map its distribution, thereby making continuous maps of species distributions. When we stack, when we combine these individual maps that we make per species, and we count how many species are there in a given site, we can make estimates of species richness by counting how many predictions are there for numerous species. And we can make these maps estimating hotspots of where are the hotspots of higher species richness, meaning hotspots of biodiversity. we can also project in space and time, meaning that we can feed the models information of future climates and assess changes on species richness, but also how the communities will change, how these communities will change under future climates. So as I said, the models are fed with two sets of data, occurrence records where species are in space, and also environmental information, the environmental information of the species are exposed to. So for this case of the kelps and the seagrasses and the cold water corals, we compiled millions of records. And in these compilations, we do a very important exercise, which is to prune out or filter records that are wrong, so the models only deal with information that is potentially correct. How can a record be wrong? Well, maybe someone recorded the presence of a species in a given ocean, but while pointing out the name of the species, it might included some taxonomic bias. It might mislead that information. So, for instance, a species that is only native in the Atlantic, if you go to databases where we get this data, maybe for that species that is only for the Atlantic, you may have records in the Pacific or even the Arctic. So we use the information that is published for every species, and we filter out these records to make the best information that we can on the occurrence records that will feed the models. We also use environmental data. Our team has a huge effort on providing this information, and what we do is we do this information as layers that are ready for climate modelers. For instance, we can provide a layer for those that are not in a GIS. This is like a picture, and the pixels of the picture are temperature values. So we create these snapshots globally of how temperature, for instance, is distributed, alongside with the conditions of nutrients and oxygen in the water that are important for animals and so on. So we can also, we also do this kind of estimates, or we also provide this data under future climates, under future scenarios of climate change. So we can then predict our models to the future it is. Most models, until very recently, were based on temperature only, but we incremented the availability of these data sets of environmental data, and nowadays we model with different layers, and for this particular case of the marine forests, we modeled beyond temperature, so we have for the seagrasses and the kelp information on sea ice coverage, its thickness, nutrient conditions because they rely on that, wave exposure because it can be detrimental for them, and also salinity. And for the corals, because they are animals, we have oxygen and primary productivity and salinity and so on. So we've made these models per species. We then stack them, and we found that on average, there are drivers beyond temperature that are also important. So we verified that for the seagrasses and the kelp forests, waves and nutrients have a major contribution on the occurrence of these individual species. And for the cold water corals, it was more interesting, in my opinion. They really rely on oxygen conditions and on productivity and also acidification. And we know that the ocean is projected to shift all of these drivers, not only temperature, but productivity levels. It might be more acidic in the future, and also with lower content on oxygen. So this was very important for us to verify. And the models performed well, so we have these estimates of accuracy. Basically, they go between 0 and 1, and on average, they were very good. And by stacking these individual models, we provided the first overall view of the hotspots of marine forests. So, for instance, for the kelp forest, we verified a huge hotspot of diversity that wasn't known before, with more than 30 species in this area in the Northeast Pacific between Point Conception and the Aleutian Islands. And for instance, for the seagrasses, we verified that this area called the Coral Triangle, and also the Indo-West Pacific and Northern Australia was very important with a lot of species. And for the cold water corals, the Caribbean is very important, and also this area of the Indo-West Pacific. These maps are aggregated for visualization purposes, but we provide them at medium to high resolution, meaning that per species, and for these thousands of species, we have information at 5 kilometers globally. So actually we can work on more local patterns of species endemicity and diversity. So we verified that beyond temperature, there are drivers that are also important, and these drivers might change in the future. And we provided the first overall view of where are these hotspots of biodiversity, feeding information beyond what was known before. That was towards mammals and fish species. So this was related to species richness patterns. So now I would like to speak a bit about how these climatic drivers, and mostly past climate changes, may have an effect on structuring genetic diversity at the global scale. So how the effects of climate in the past structure areas that today have higher genetic diversity. So they are evolutionary hotspots. So there is a paradigm also in terrestrial climates of the way these past climates structured genetic diversity, meaning that during during glacial periods, like the last glacial maximum, 20,000 years ago, the advance of these massive ice sheets covering most coastal and continental land masses led species that were distributed in higher latitudes to contract their ranges to lower sites, lower latitude sites. During interglacial periods like the mid-Holocene, which is called the climatic optimum, 6,000 years ago, or even the Anthropocene that we are experiencing nowadays, the warmer climates and the retreat of ice allowed species to recolonize higher latitudes. In this process, extinctions might have occurred and are occurring at lower, warmer temperature range margins. So these range shifts of glacial periods and interglacial periods structure these areas that have optimal climate conditions for species for the long term that we call climatic refugia, where populations were allowed to persist for long term and accumulate diversity by random mutation. Diversity, genetic diversity, of course. While where extinctions or expansions occur, we expect or we measured in the field lower genetic diversity due to bottlenecks, meaning that populations get lower effective sizes, and also these founder effects. So basically founder effects are these effects that during the colonization from refugia, where you have a lot of diversity because of long-term persistence, this colonization of the higher latitudes is made by few individuals, meaning that few individuals mean few alleles, few genetic diversity. So in the end, while they propagate and colonize these higher latitudes range margins, we end up with very homogeneous, low diversity areas in post-LGM effect areas, while we have in refugia a lot of genetic diversity, okay? So this paradigm is well accepted. However, until very recently in the marine environment, it had no replication at the species level. So, and this is important because if we look at how these glacial periods in the last glacial maximum impacted the higher latitudes, we see that between hemispheres, we have contrasting opportunities for evolution. For instance, during the last glacial maximum, the North Hemisphere was impacted until Brittany or this area in Maine or down to Vancouver Island and all the Aleutians, while in the Southern Hemisphere, besides Antarctica, only Patagonia was affected. So this might bring contrasting opportunities for evolution. So we used one species, Macrocystis perifera, which is this amazing macroalgae that can grow to dozens of meters. Why? Because it is distributed globally and has this anti-tropical distribution with two range margins potentially exposed to the effects of the advances of the ice of the last glacial maximum. So what we did to test if for the same species we have the same signal of past climate changes, what we did was we estimated genetic diversity across its range with hundreds of sites from Alaska in the northern hemisphere toward the Southern Ocean. And then we compare these estimates of genetic diversity with independent species distribution models. So we've made a species distribution model for the species, and we predicted the distribution during last glacial maximum and present day to infer regions of extinction, but also regions of persistence, these areas that we are interested, that are the refugial areas for the species that might allow the accumulation of diversity. So here just two maps showing present day predictions of the models and also last glacial maximum predictions of the models between the two hemispheres, being in red the suitable habitats for the species. And when we compared the two maps, we verified that there were regions with very high range contraction due to the advance of the colder climates and the ice sheets during the last glacial maximum. Here, between the Aleutian Islands and this area in Canada, we also had contractions in Argentina and throughout all the Southern Ocean. And when we compare the potential extent of these species globally, it contracted 40% of what is known today. So the range, the distribution of the species was much narrower. However, despite this contraction, we verified huge areas of refugia. For instance, from Vancouver Island to Baja California in Mexico, from Peru southwards, and throughout we call the circumpolar currents. So along the Antarctic area. So what we did to compare these effects of past climate change with the genetic diversity, we compare the distance of the populations to the refugial areas in the assumption that inside refugia, we estimate higher genetic diversity. And we verified that as you go and move off a refugial area, here in kilometers, you have a huge decrease in genetic diversity. So if you move outside just 75 kilometers from our predicted areas of refugia, you have threefold less genetic diversity, meaning less number of alleles. And we also counted the endemic diversity, meaning alleles that only occur in a given population. This information is very important for evolution because it's unique information that is not distributed anywhere except in a given population. And we found an even drastic scenario, that is, if you move 50 kilometers from these refugial areas, you have less than one private allele on average, and if you move more than 150 kilometers, you have zero endemic diversity, and we verified that from the 150 populations that we sampled, 50 had no endemic diversity. So this study allowed to corroborate the hypothesis of past climate changes, but with replication across ranges. So this was the first study that had two ranges exposed to potential similar effects of past climate changes. So in higher latitudes, we got low genetic diversity due to this expansion of few alleles after the effects of the last glacial maximum. And in the lower latitudes, today warmer, it retained what we have in the hotspots of genetic diversity for these species. So these areas are linked to the ability of the species to evolve in the future. These areas are very important. Okay, but that was a single species example. So throughout our research goals in our team, we asked, okay, so that was a species example, but where are the hotspots across marine forest species? Where are regions in space that consistently have higher genetic diversity linked to higher adaptability potential? So this information is completely limited. Until this study that we've made, there was no overall view for any marine group in the marine environment. The information was scattered between areas or species or interests that people have on their own taxonomic species or focal species, and there was nothing there. So we thought about, because the information is scattered, we thought about making a unified model, meaning that a model that combines the scattered information between species and regions and incorporating a unique model that could be transposed across the ranges of hundreds of species. So we used a machine learning model fitting genetic data against proxies of exposure to detrimental conditions, past conditions, but also contemporary conditions, because ongoing climate change may also have an effect on genetic diversity levels. So what we've made was to compile all the information that was available in terms of population genetic diversity for this group of brown macroalgae. So we covered 25 species for 600 populations. And what we did was we made this unified model founded on the niche centroid theory. So the first thing was to define the niche centroid of each species. This niche centroid is a theoretical point, okay? But basically we expect there, because it's the center of the niche conditions, the niche can be gradients of temperature, gradients of ice, gradients of nutrients, etc. So the center of this point, we expect the conditions to be optimal, not marginal for the species. So therefore, individuals have higher fitness, and if they have higher fitness, they will have higher abundances in their populations, and therefore higher genetic diversity, in the detriment of lower population sizes, very residual populations, where we know that genetic drift has a strong effect. Genetic drift is the random purge of alleles. If you have very low abundant populations, it is likely for you to lose alleles because those alleles are not well represented in the population. So then we define this niche centroid in the multi-environmental space across each species. So here we have the range of a species. Across this range, we have the environmental gradients that the species is exposed to. For instance, in our case of the marine forest, this was represented by temperature, salinity, nutrient conditions, and ice. And then we found the center of this multi-environmental space. And because the centroid is well positioned in space, in this environmental space, which is multidimensional, because it's centered in a space, we can calculate distances, environmental distances between each point and any other point. So these other points can be populations in space. So they can be the populations that we sampled for genetic diversity. So we can sample how dissimilar climate is in a given population compared to the optimal of that species, to the optimum of that species. Here an example for a given population that is sampled, and we have used, as I said before, two contrasting climate exposure indices. One, contemporary conditions, and so the distance to contemporary climates, and also distances to how the populations were exposed in the past. So you see a given population in the field, and you ask, what were the conditions in the last glacial maximum that this population was exposed to? So we had these distances, here the distance to the niche centroid of a given population, here less central than this population as an example. And we also made the same exercise with the conditions that was exposed to during the last glacial maximum. So did the model fitted this information? In the end, we combine all populations for all species that we sampled. So we had genetic diversity values against these distances towards optimum of climate conditions. And of course, we had two competitive drivers. We asked, is it present-day conditions or past conditions? So we fitted the model, and in my opinion, we had a very good model, considering that the populations were scattered, the species were different, and we were expecting effects from niche centroid theory proxies. So it explained very well. 80% of the data was explained in this model. And the last glacial maximum conditions explain 7 times more data variability than the contemporary conditions. So again, by combining all the information, we verified again that what is important for what we perceive today, it's the effects of past climate change. And because the model was unified, we can then predict per species. So then we had a model where we predicted for almost 300 species. So we stacked these estimates, and then we verified, okay, for the group of the kelp forest and this brown macroalgae, the marine forest, where are the hotspots of higher genetic diversity? And we found, again, the Northeast Pacific, a very potential evolutionary hotspot, and then the Northeast Atlantic, here the Bay of Biscay, very important towards the British Isles, Southern Australia, New Zealand, and Southern Japan. So this was the first overall view for any marine taxonomic group that we provided estimates of genetic diversity across species. And again, these maps are coarse here, but we provided at moderate to high resolution, those 5 kilometers that I said before. Then we make this comparison, which is interesting in my opinion, especially for stakeholders, and if you want to have an overall view of the important areas for these groups, that is making this rule of having this map reclassified to, okay, where do I see higher species richness and higher genetic diversity? So we had a moderate spatial consensus of these areas, and there are hotspots that are very important from genes to communities. So these were the effects of past climate changes on genetic diversity, but a major constraint in the ocean that can really shape genetic diversity and also in the terrestrial environments is dispersal potential. So in the ocean, we have massive ocean currents that may promote very sharp dispersal barriers, meaning that populations cannot disperse elsewhere, or in the opposite, they can promote long distance dispersal. There are events recorded in the ocean for dispersal on the orders of hundreds to thousands of kilometers of individuals promoted by these ocean current patterns. So these ocean current patterns, they may erase the effect of past climate changes, because even though you have centers of hotspots driven by past climate changes, you may dilute that information, for instance, if you promote population interchange of genes. So past climate changes might not be sufficient in the ocean to explain genetic diversity. So we asked the question if genetic diversity is also explained by ocean current patterns. So what we did was we made a biophysical model, which I will explain in a moment, but basically it infers how the sites along the coastlines are connected by these patterns of ocean currents. And then we compare these probabilities of connectivity between all sites everywhere in the world with estimates of genetic differentiation, not the levels of genetic diversity, but asking how different are the populations. Because if they are not connected, they will shift through time and they will be different because they are suffering the effect of a barrier, okay? So basically what we did at the global scale, we generated at 5 kilometer resolution source and sink sites, meaning that these are locations that will, from where these particles will be thrown to a virtual ocean. And these particles, they represent, for instance, spores of kelp or larvae of corals, or even fruits of mangroves. And we track them in this virtual ocean for 2 decades with daily data on ocean currents, but this by satellite imagery. So this is actually not completely empirical because there are models there, but it's most of the data, it's observations. So, and we track the position of each particle every hour. So this was a huge computational exercise with billions of particles everywhere. And every time a particle that was drifting in the ocean reached another location, we assigned the connectivity event, and then we could calculate probability of connectivity by just summing, for instance, how many particles from Northwest Siberia got to Brittany, divided by the number of particles that were released by Northwest Siberia. So we have estimates of probability of connectivity between all sites. And then we needed the genetic differentiation data. And we used 2 groups that are completely contrasting in terms of their dispersal potential. We used kelp forests and brown macroalgae, which are very low dispersive because their spores can stay in the water up to 5 days. And we contrasted that with another group of mangroves for which fruits are known to raft in the surface of the ocean for dozens to hundreds of days. Okay, so we compiled genetic data, this differentiation data for mangroves and also for the macroalgae, and then we made these species by species models in the attempt of answering does lower probability of connectivity explains lower, the opposite. Does increasing the connectivity between populations reduces genetic differentiation, right? So if they are well connected, their differentiation will be lower. So we answered this question species by species. So our biophysical model, here an example for Cistozeiro, which is a brown algae, which can raft in the ocean. These are the populations of this particular model that were sampled. So with the biophysical model, we have their probability of connectivity between all sites. And then with this information, we predicted genetic differentiation. And for this example, for instance, we had a very good agreement between predicted genetic differentiation and observed genetic differentiation. Another group for another brown algae, a good agreement also. And then here, an example for a mangrove. And what we found was that regardless of the group, meaning that regardless of the dispersal potential of the species, kelps versus mangroves, regardless of the species, so comparison between species, the region of the ocean, and also the genetic method used, which really impacts your estimates, we found that connectivity driven by ocean currents helped explain genetic differentiation on an average of 45% of data variability. So we found that ocean currents are very important in what we observe in the contemporary levels of genetic differentiation across all of these species. So this was very relevant, of course, for our biogeographic questions and also for evolution, of course, because barriers are important because they create differentiation. So for evolution properties and also for conservation. And I'll go talk about that in a moment. So genetic diversity and differentiation also signaled by the effect of long-term patterns of ocean currents. Okay, so I covered species richness patterns, genetic diversity patterns, which are a result of past climate changes and also patterns of ocean currents. And now, of course, the question, we have the patterns. So if we shift species distributions under climate change, what will be the results for these communities and these different groups that cluster into what we call marine forests? So until now, or until very recently, there was no impact assessment of how climate will make an effect on these groups, like the ones that feed IPCC. So we used the machine learning models that we had species by species, and we fed them the scenarios of the shared socioeconomic pathways for end-of-century conditions, and we covered from very low emission, meaning sustainable scenarios, which we expect will be low impact on biodiversity shifts, and we also covered towards fossil fuel development scenarios with very high impact on biodiversity. So from the Paris Agreement expectations towards higher emission scenarios. What we found was that, of course, regardless of the group, regardless of the species, higher emissions are linked to increased species richness, losses, and changes. But the question that we always ask, okay, we have different scenarios. We know that increasing emissions will increase changes in ocean drivers. In the later instance, it will shift biodiversity patterns. So we ask, but are we towards sustainability or more aggressive scenarios of greenhouse gas emissions? So what we know from observations, and this is from Copernicus Marine Service, is that we are already surpassing the targets placed for end-of-century conditions. The 1.5 to 2 degree conditions that is the major goal of the Paris Agreement are being surpassed mostly in the recent years. For instance, January this year had an average sea surface temperature, ocean and terrestrial, of 1.75 degrees above pre-industrial levels. April 25 was the 20th consecutive month above one of the milestones, which is the 1.5 degrees, and the last year averaged 1.6. If we want to reduce these conditions to meet the Paris Agreement targets, climate researchers point to a drastic reduce in carbon emissions that we need to comply to the levels of the 70s until 2045. So based on observations and the trajectories that we are not aiming at, some climate researchers are at least leaving sustainability scenarios. So what does this represent to marine forests? So under higher carbon emissions for the kelps and the seagrass, what we have is very drastic replacements and shifts in species richness. So here green means higher species richness, here purple means, or darker purple means lower species richness. So what we'll have is the expected poleward shift of these communities, okay? Both in kelps and seagrass. Corals, because they live much deeper, some species can prevail at thousands of meters depth. We also have this pattern of poleward shifts, mostly from this area of the Coral Triangle in northern Australia. But what we have cross species is depth shifts. So they might accommodate their optimal conditions by moving to deeper waters. But one thing that I like to do in my research and my presentations is not only to show the drastic effects of climate change. Despite these trajectories, we have huge opportunities for conservation because cross species, we have high degree of refugia, meaning that this map, for instance, show that if you are closer to one, it means that all species will prevail there. So despite reshuffling, at higher latitudes, we see a lot of potential to conserve species richness hotspots. So for the kelps and the seagrasses at higher latitudes, and the seagrass at this area of the coral triangle, and for the cold water corals, what we see is that refugia long-term persistence is globally distributed, however offshore, meaning that you have to go to deeper waters. So coastal areas will be highly impacted. So we had the estimates at the species level, but we also had the information of where are the hotspots of genetic diversity. So the trajectory of higher emissions may be particularly detrimental to these areas where we can see or where we predicted more than 20 species having populations with high genetic diversity. So these are the two main areas that might really lose their evolutionary ability. This in southern Australia and also southern Japan. Just here, a case for our Iberian Peninsula is one of the most impacted areas in terms of genetic area. So, in terms of genetic diversity. So just a very brief point on conservation priorities. So we, as I stressed, provide these maps at the highest resolution that we can. So we have projects ongoing where we feed these models and these predictions to try to assist on the prioritization of marine protected areas. So we are framed, I think most of you have heard, the post-2020 Global Biodiversity Framework. It has this huge milestone of conserving 30% of the ocean through well-connected, climate-resilient networks of marine protected areas that should focus on biodiversity. So how can we assist this? Richer biodiversity by providing these maps, of course, where communities are richer. And here, of course, this is the large-scale aggregation, but we can zoom in at the Iberian Peninsula and compare which areas across, for instance, the Mediterranean coastlines of Spain, which areas have higher species richness of these groups. Also, the genetic diversity information, climatically resilient network of marine protected areas. So to be climatic resilient means to be able to sustain climatic exposure in the future. So these refugia estimates are key because these are areas that, despite trajectories of aggressive climate change, they still prevail or sustain most of their species richness diversity, and also well-connected. So we are using these biophysical models, these probabilities of sites along coastlines being connected, to identify regions that are more central, meaning that if you want to travel, for instance, from South Brazil to this area of the Western Africa coastline, if you want to go from one point to another using the ocean currents, because this is what marine species need to use to make their trajectories, at least these marine forests, we identify these areas that are stepping stones. So if you want to cross oceans, you need to use these stepping stone areas. So we map these regions that are central, meaning that they are stepping stones. So they are more important because if populations need to migrate under climate change, or if populations want to exchange their genes, they need to use these sites that are more central in their distribution. Okay, these are baselines that we try to incorporate in the assistance of marine protected areas. How do we do it? By complying with FAIR principles. So all of our climate data is published in terms of describing what do they represent and how they are made available in permanent repositories. All our recurrence records that are pruned, meaning those records that are wrong, we flag them and we provide this in data papers of species distribution that are, in our opinion, better than going to major biodiversity facilities. We also publish our range maps because despite the usability of, in our questions of climate change effects, of where are the hotspots, etc. We also feel that this can assist other questions. So the predictions per species across scenarios of climate change, we also publish them. And the connectivity at this moment is also being published. So we believe we have at least a contribution to the designation of marine protected areas. So this is our humble and small team. And all the articles that I talk, all the software that allowed us to do these estimates, and even the data that I talked, everything is available at biodiversitydatascience.com, or please just send me an email, any question, please. Thank you so much. I hope I complied more or less with the time.
You did. Thank you for your excellent talk. We have time for questions. Shahar?
First of all, let me say, wow. Yeah, for me, it was very impressive. I know that handling, for instance, currents in the ocean is not an easy task, and understanding connectivity in the ocean is something that is a huge challenge. So it was very impressive to see all those studies. I had like a general comment that can be maybe be more detailed, but I was wondering, how much do you think that those models represent the reality? For instance, I can say different points like, can some of those, I used to work with animals, so I don't know the really definitions for plants, for instance, but those spores, for instance, of the macroalgae, can they really move those large distances in the open oceans, for instance? And the second question, maybe for the Mediterranean Sea and the Eastern Mediterranean, I've seen like from the data that you showed that there is, it seems to be like more coral species than macroalgae. So I wasn't sure, because I know it's, for what I know in the Eastern Mediterranean, it's not usually the case, what it might represent. Yeah, so.
Thank you so much. So, regarding the potential of these species to use ocean currents, the thing is, this is why I like marine forests also, because they are passive dispersal. So it's very easy to track these signals because they can only move by ocean currents. And of course, that spores travel for very few days, but all of these species make what we call a raft. It's an entanglement of a piece of a species. And if it's in its reproduction season, it can actually move away from its origin location and actually then release its spores and make a new population. Actually, this topic, when I started to work with it, I was more interested at the large spatial patterns, because this is what we can really track, like the sharp barriers and the opportunities for long distance dispersal, which then we can relate with the differentiation of the populations. But because there is a lot of investment this last decade on these species, there are a lot of ecologists tracking expansions. And actually, the estimates that we originally made on the potential of these species to expand their ranges are more or less what they are perceiving in reality, in records, under climate change, mostly along the continental coastlines of Brittany and Cornwall for the species that we are mostly interested in Iberia Peninsula. So this to say that in my head at the beginning, this was very theoretical, and trying, as I do last glacial maximum, I was looking at the bigger picture, but actually this has a good representation, in our opinion, of reality. So I will go to the second question just to say, yeah, this has a good, I remember something, but then I got lost. So it has a good representation. And yes, and we are integrated this in future range shifts because it might be of interest. So your second question was about?
For instance, the Eastern Mediterranean Sea, that you show that there are some a lot of, seems to be like relatively a lot of corals with respect to macroalgae. How can it be?
Well,
what is the data that was used? Maybe it's only a portion of
I mean,
species.
This was global data. And actually, that is our perception of the Mediterranean. So brown macroalgae do not prevail in low nutrient warmer waters. Okay, so we have, for instance, refugia of species that are very cryptic of this large brown macroalgae that are, for instance, in the Strait of Sicily, very deep. Compared to the Atlantic, where they are everywhere, we have records that are very punctual, so they are not distributed in the Mediterranean, but these points in space that are very deep, so the water is transparent enough for them to receive light. And there are conditions of nutrients because, for instance, the Strait of Sicily has a lot of resuspension of the water. And when we go and we sample genetic diversity in these populations, they are the highest diverse populations. So if we look at the snapshot of the Mediterranean, brown macroalgae are here and there. But then, when you measure genetic diversity compared to the counterpart of the same species in the Atlantic, it's much more diverse, meaning that in colder periods, like the last glacial maximum, maybe the Mediterranean was very abundant in marine forests. So our perception is, okay, if you think about Sargassum forests and Schistosera forests, they are distributed in the Mediterranean. But if you compare species richness of brown algae of the Mediterranean with, for instance, Northwest Iberia and Brittany and British Isles and even Norway, in our comparisons, in what we perceive in the field, there's much more higher diversity across these areas, much higher. Regarding corals, I'm not an expert in corals. This is a very particular group because it's not transversal, it's cold water corals, meaning that it's fun corals and those that are more into deeper waters. Again, why we use this group? Because they are easier to model, because they are not distributed in the hotspots of today's warmest climates, for which it was very hard for us to infer future effects of climate change. So we work with mostly with cold water corals because of this. And again, we know many populations that are luxurious and much more abundant across the Mediterranean compared to the Atlantic. So, I don't know. I'm a macroecologist. I really like to work with data science, and I really like the inputs of the experts. As I move, I feel that I'm not an expert on biology. But we try to make our best with our models.
So I was thinking on whether you could, when you started talking about the kelp forest, whether you could, I mean, the potential to make estimates on carbon storage and sequestration in this forest, right? I don't know how much work has been done on this. I guess it's pretty challenging. And I was guessing if you have data, or if it's possible to gather data on height of kelps. I mean, that exists. And then, how deep are these forests? I mean, is it possible to look at them or kind of identify them with remote sensing data or not really?
Okay, two very interesting questions. So regarding blue carbon, I'm not referring to it here. I collaborated in the first global estimate of blue carbon driven by this group of marine forests, the kelps. So what we did, we had cores of the carbon estimates in the sediments across the globe. And there was a very brilliant data scientist, because he's not a mathematician, but it's more like myself doing these models, that integrated in this biophysical model the potential for the rafts of the algae to sink and trait information between species of which species are able to sink compared to others, because there are some to have facilitated dispersal. They have these vesicles of air bubbles, so they cannot sink, period. Why the sinking process? Because for you to sequester carbon in the ocean, you need to go to a depth that is sufficiently deep for not to the wind-driven resuspension to bring the carbon back again. So there is more or less this estimate of 200 meters as an average for the ocean. So we had the global model from where particles were released from the coastline, and we measured retention and exportation, And while exporting, we had a mathematical function on sinking, and every time it crossed the 200 meters, it was signaled. And this was then cross-related with the cores. So we had a very good agreement, and we had the first estimates on, for instance, the global potential of these macroalgae to sequester carbon. So send me an email, I'll forward you the paper. It was, in my opinion, a brilliant paper. It has a huge echo on newspapers and etc. But I got a bit frustrated. You know why? Because the yearly potential of that we estimate, it's an estimate, of course, but the yearly potential was around the number of cars of Spain. So I felt that, I mean, these species are distributed globally. I mean, if you go to a temperate, cold area, you put your mask and you see. I mean, sometimes you don't perceive as a marine forest, but we group everything. It's a kelp, it's a brown algae, they're there. I mean, they are globally distributed, and per year they can only capture, in my view, I was expecting it to be like marvelous species. They're marvelous, but I mean, the amount of carbon that we emit, it's a lot. I mean, because the amount, it's just the number of cars in Spain, and you take everything out of the equation that we do globally, right? So, and the other question, which was very good, was about
The height.
Of course. So I have a paper that I done with using convolutional neural networks, but those that have mask recurrent convolutional neural networks. So basically, they do not only predict it's there, something in the image, actually they make a mask. So we used Landsat imagery, and we trained the model with macrocystis, this large brown algae, and the model went very good. So we then hindcasted 30 years of Landsat across all the Northeast Pacific, and we had estimates of abundance, I mean coverage, not abundance, but coverage, which matched very good the El Nino periods. But major issue is that to use remote sensing, it's on macrocystis. Because if you think of any coastline, looking from above, you cannot perceive much. Right. So these algae are huge, and they are perceived from space. I have a very nice contact, Kyle Cavanaugh. He's working actually in my team now. He's doing his sabbatical for 1 year. And he's using this latest resolution satellites that I'm not aware, but he talks about 3-meter resolution. So he's actually exploring connectivity at very low scales and collapses at very low scales using this data. So if you want, I can forward the contact. He's the person.
Okay, thank you.
Yeah.
I think we may have this question and then another one, and we unfortunately have to close.
I'm very happy that I can have the last question. First of all, I want to thank you very much for being here today. And I want to thank you very much because the second strongest name in your group is a Greek female scientist, which is very good.
She's brilliant.
She's brilliant, yes. I know her from the literature we discussed before. So my question is, I had this question, and I was having this question since I was working with marine systems. And I think you are the right person to ask this question. When we see these marine global maps, these biogeographic patterns, a naive eye would say that we see huge gaps in the oceans. We have huge regions that, of course, are undiscovered. And my eye 2 months ago was that just 2 months ago, only 27% of the oceans have been discovered, have been scanned. So my question goes to the fact that we see that these patterns are on the continental shelf, are very close to the land. So do you think that since you have developed the infrastructure, all these models, you have all this information in this part of the ocean systems, do you think that if you try to incorporate land-sea interactions, for example, hydrological extremes or meteorological extremes, but in the land that are connected with the continental shelf, do you think that has a value? Do you think that it could be the next step in your models? I would like your thoughts on that.
I think the next step is completely integrative, although sometimes we are challenging by our time. we can understand that there's a major potential driver for my process, but sometimes there are constraints there. So regarding the data limitations, at least we are working with coastal species for which we know from very old biogeographic descriptions, etc., their global distribution. For instance, kelps and seagrasses, they are very well described, cold water corals less, but for instance, we didn't publish yet the global distribution patterns of sponges because the data is very, not scarce, but very oriented towards where you have major institutions providing the data. Okay, so that's the first thing. As deep as you go, because sponges are very deep also, the deeper you go, the less you know. And when you say 20%, that 20% is exponentially represented in the deep, okay? So we are confident that our models for the seagrasses and the kelps, for instance, represent the potential distribution, not the realized, for these groups, okay? So that's the first thing. The integrative approach, it's very interesting because imagine the case of Portugal. We have sometimes periods of huge fires. So basically you have this wash off of these very dark sediments that we know from field surveys that cover the layer where the spores of the kelps are waiting for recruitment for the next season. So we know that it has a huge impact, at least when you go to the field. But at the scales that we are talking about, no one has made it before, and is a major question how these connections between terrestrial and marine environments explain the drivers of their distribution. There's a lot of temporal data for kelps. For instance, we are using temporal data to model abundance globally for kelp forests. So maybe the inclusion of that interface would be very interesting, with proxies, for instance, of river runoff or something like that. And the attenuation of water, because from satellite, we have how the turbidity attenuates the penetration of light. Actually, we have this sensed remotely by satellites. So you have good estimates to try to compare.
I have mainly in mind the freshwater ecosystems, like deltas and brackish systems. I had in mind freshwater ecosystems, like deltas, like for example, we could potentially assess the genetic diversity in freshwater ecosystems and what's the vicinity with the genetic diversity in marine ecosystems, because we know that deltas are quite rich. So from the terrestrial point of view, I had in mind the freshwater systems, but I'm covered with your...
Okay. Any quick question? If you have? Okay. So that's the last one.
Thanks for the talk. So we have a number of studies, mainly on vertebrates, showing that genetic diversity is not necessarily correlated with extinction risk, simply because you're losing genetic diversity that is not related to traits such as reproduction, dispersal capability, etc. So I was wondering whether those endemic alleles you were talking about, are they related to key functional traits in marine forest species?
Yeah. So that's a huge question that my previous colleagues, because I diverged from a research group that was completely towards genetic diversity estimates and actually laboratory experiments, physiological experiments. And I was a huge outlier, so I made my path. But actually, that's what they're doing now. And for instance, a very recent paper of them shows that they are not correlating with levels of genetic diversity directly, although I know that those are the richer populations across the range, but they cross-related that there are unique alleles that express physiological protection during marine heat waves that are conserving, in a way, the low latitude populations of these species that are being exposed to the more detrimental conditions. So they are making common garden experiments. They are bringing the organisms from higher latitudes They are working on the first generation because they don't want the organisms to have the story of what they have been exposed to. So they make the first generation in lab, and then they expose this first generation and see gene expression and physiological tolerance to warmer conditions. And for the species that they are working, there's a correlation. However, I've never said, and I think there are traits, there are multiple traits, and there are some that can really increase against extinction risk. I only reinforce the fact that it's evolutionary potential. I mean, higher genetic diversity is related to evolutionary potential. Not, in my opinion, you may have signals for some species, but it might be that for some, you may be a lot of genetic diversity, but then you collapse doing a marine heat wave event, compared to other that has lower, but has a particular trait that may, yeah.
Well, thank you, George. It was a really stimulating and well-integrated story.
Okay.
I hope you enjoyed. This talk is going to be stored in the museum repository of seminars, so you're welcome to see it again if you need. Thank you very much.
Thank you.